Then is the light microscopy. First of all, we will study about the light microscopy. What is the light microscopy? Light microscopy refers to the use of any kind of microscope that uses the visible light to observe the objects or to observe the different specimens. Which will use by the light. We can use it by the contrast way, or we can use it by the different ways, but it will use the different beams of the light. From the light microscopic point of view, we have different classes of the light microscope: compound light microscopy, dark field microscopy, phase contrast microscopy, or differential interference contrast microscopy. Similarly, the fluorescent microscopy and the confocal microscopy. These are different kinds of the microscopes or microscopic point of view. From we will study them one by one, that what is the compound light microscope, how it is used, and what is its principle, and how it can be used by the different ways and different objects. First of all, we will study the compound light microscopy, and we also call this microscope is called the compound microscope. Light rays from an illuminator which are released from the light, and this is called the light source, and light source is an illuminator. The light passes through the condenser, we will see in the next slides, and then has the lenses that direct the light rays through the specimen. From here, the light rays pass into the objective lenses, and the image of the specimen is magnified again according to your desired ocular lens, the eyepiece, and then you can easily see that what kind of specimen is this, and how much the magnification is required by the personal interest. Magnification, we will see the two different kinds of definitions here: the magnification and the resolution. Objective lens X eyepiece lens is called a magnification. It is a ratio of the size of an object seen under the microscope to the actual size observed with unaided eyes. It does not affect on the physical size, but its effects on the actual size observed by the unaided eyes, and it will be called the magnification. And magnification can be, there is the magnification knob, and you can just change your magnification according to your desired regulation. Then the resolution refers to the ability of the lens to distinguish two points that are specified and they are distance apart. The resolving power of the human eye is 0.25 mm, and the light microscope can separate the dots that are 0.25 micrometer apart. So in this way, the resolution is make it the object make very clear, because the resolution power of the human eye is 0.25 mm, but the light microscope, it can separate the dots even in the 0.25 micrometer apart. The electron microscope can separate the dots that are 0.5 nanometer apart. Even the electron microscope these days, the very specialized kind of microscopes are available, and they can separate the dots that are 0.5 nanometer apart.